want to talk about microbiomes, but before that, I would like to thank you for staying and thank the HFSP for the funding. So as I said, we're going to talk about microbiomes, and the microbiome and microbes are extremely, extremely important for our planet. They are, and for us, they are involved in biogeochemical processes, in everything that is related. I want to move, but it's not working. It is working. Okay. Everything that is related to health and disease, and also for sustainability. But when people typically look at microbiomes, we usually look at a list of microbes and the functions that they have. But what I want to argue is that it is way more than just whoever is there. It's also about how these species or strains interact with each other. So interactions is essentially everything. Even if you're looking at or interested in a specific bacteria, let's say it's a pathogenic bacteria, you're a doctor, this bacteria forms part of a larger microbiome where it interacts with other bacteria, it interacts with other parasites, and also interacts with subcellular entities like plasmids. So plasmids are double-stranded DNA elements that are infectious. They move between bacteria in various ways. So here I'm presenting conjugation, and this is one way. And on their way, on their movement, they also take with them genes that can be later transferred to the microbe. And this is really, really important because on the one hand, plasmids are infectious, so they're costly. But on the other hand, the genes that they have, the accessory genes, can provide the microbes with some advantages. For example, resistance to antibiotics, or maybe enhanced metabolic capabilities, or enhanced virulence. So because this is so important, there's been tons of research into plasmid bacteria interactions, but typically on a one-to-one scale. So it's one plasmid, one bacteria, maybe a few. But in nature, these entities they form huge networks of interactions. And it is the structure of the networks that determine the dynamics and eventually determine the function, such as resistance to antibiotics. So we have a largely open question, which is how does microbe and plasmid network structure affect community dynamics? And this is a really, really difficult question, especially if you want to understand it in the wild. So you start simple. We started not in the wild, but with modeling and experiments. And because this is a large question and difficult question, it is also a interdisciplinary question. It requires a team. So our team is composed of Jamie Hall, who is a microbiologist in University of Liverpool, along with his postdoc Caitlin, who is, by the way, here, and she will give a poster. Professor Manuel De Domenico, who is a physicist in Padova, with his postdoc Johannes. And on our team, we have Ingji, who is a postdoc on my team. And together, we have these microbiology, physics, and ecology disciplines working together. So when we started looking at this, we started from a conceptual shift. We said, okay, it's networks, but it's not only a single network. There are multiple kinds of interactions occurring at the same time. There is infection, which plasmid can infect which bacteria. We also have compatibility, which means that not all even if plasmids can infect the same bacteria, they don't necessarily are able to co-reside in the same cells. So they have to be compatible. And there are also barriers to horizontal gene transfer. So what I'm going to show you today is actually focusing on these two networks. This is still ongoing, and I don't have a lot of time. So we wanted to understand via a factorial design what is the effect of structure on dynamics. And so we selected a few structures for the compatibility network and for the infection network. And these structures are inspired by disease ecology, because I'm a disease ecologist by training, and these are infectious agents. And we have three bacteria and 4 plasmids. And you might say, well, this is really small. You just said, nature is diverse. And yes, it's diverse. And when I first approached this, I approached this from like an ecologist. I was like, okay, simulations. We have 100 species, 1 million plasmids. It was just so complex. We couldn't figure anything out. It was really, really difficult, slow to run. And the physicists were like, whoa, you should stop. Let's go the physics way, strip it down, very simple system so you can understand what is going on, and then maybe you can build back up. So that is what we did. This was a great insight. So together with the physicist, we built this model, which is based on this structure, and we have identical strains for now with stochastic simulations. And these simulations, they run in time, so you have a time, and then on the y-axis, you can quantify prevalence or abundance of bacteria. And what I'm going to show you next are the results from the end of those simulations. So if we start with just the control experiments, no structure at all, then you get essentially what you expect to get. On average, each of these bacteria was dominating the whole community, outcompeting all the others, and the plasmids reached more or less the same prevalence. But what happens when you start adding structures? So the first cool result was that when you structure the plasmid networks, then you actually affect the bacteria. Because here on the this is the fraction of simulations in which bacteria live in coexistence, so 2 or 3 bacteria together. And you can see that when you have no structure, then very quickly only one remains. But when you add structure, the transience of the coexistence is much, much longer. So you directly affect bacterial dynamics just by changing the structure of the plasmid networks. On the other hand, when you change the infection network, then this affects the prevalence of the plasmids. So instead of them having same or similar prevalence, now there are 2 plasmids dominating once you have a clustered network. And you can also have multiple structures at the same time. So the first phase is what I just showed you, but then you can have 2 structured networks, and then we get something completely that we did not expect, and suddenly one plasmid is overtaking all the community. And that shows us that these coupled networks, there is interplay between these networks that produce non-additive effects. But okay, this is all nice, but it's just a model, so is it relevant? So this is where Jamie came in and Caitlin with the experiments, and we asked them to engineer. we said, okay, we're going to engineer the networks. And it turns out that it's really, really hard to do these experiments. You work with the bacteria. Caitlin can tell you all about it. You work with the bacteria. They do whatever they want to do. They don't ask you. They evolve. They compete. Oh, my God. I'm staying with models. But what I want to show here is the difference between, or what we can understand between the difference, the modeling and the experiments. So they did it, and they made this experiment with a modular network that you can see here. And here are the dynamics of the system. So this is the green bacteria, and this is the blue plasmid that is dominating. And what we did first was to take the model and match it to the experiment. So now we reparameterize the model according to the experiment, and we qualitatively got the same results. And then now that we had a model that was based on a real system, empirical system, we could again go back and play with the model, and we changed the network structure here. This is a different structure, which completely changed the qualitative dynamics of the plasmids. So this is really cool second line of evidence for the fact that the plasmid spread and dynamics is governed by coupled network structures that generate these non-additive effects. Well, okay, these are very small microbiomes in experimental systems, but in nature, these microbiomes are way bigger, and they occur within hosts, or so either an animal host or maybe a human host. And these hosts, they occur in the environment, and sometimes the microbes occur in the environment. So there are all these scales. And so the question is, if we can actually quantify, if we go up one scale or multiple scales, can we quantify this association between environment, host, and microbiomes? And here you can't go doing models. So this is inferring from very messy data collected in nature. And there is a leading hypothesis in disease ecology that actually is designed to address this question of the association between host environment and microbiome. And this is the disease triangle that states that the infection that you see here is results from the interplay between host traits, the environment, and the parasites, or in this case, the parasites and the microbiome. So 2 of my PhD students led this work, and so now you need a system where you can actually measure all these things. This is not trivial at all, But we have a very cool system in Madagascar where people live along this land use gradient, and they use all of it from the primary forest all the way to the village. And it's not only people, it's also other animals like rats. And there are a lot of parasites in those rats. And this is important because this interface between people and the rats and the environment and the parasites really leads, because of their lifestyle, it really leads to possible emerging infectious diseases. Okay, so the pipeline went like this. We went to the field and in all these land uses, and we captured rats, and for each rat, we had the gut microbiome that includes the parasites, so we could build the networks that you see here. And we also measured host traits like biomass and so on, and the environmental features around, so the environment. Then we built a predictive model that allowed us to predict interactions, but that model also had the explanatory part that could quantify the importance or the relevance of each of these environmental or host features. And it turns out that, yes, the obvious is there, which is good. The body mass really predicts a lot of the infection, but also the environment. And not only that, 4 of the main features that explained or infection or were associated with infection were 4 families of bacteria. And so this is one of very few evidence from the wild for associations between bacteria and parasites in the microbiome. So parasite infection is associated with host-related features, including the microbiome composition, if we look at these scales. So if I want to put these 2 parts together of the talk, the main take-home message is that to understand microbiome function, this is really hard, and it requires studying interaction networks, first of all, and it can only be achieved when you look across scales, across methods, using multiple methods, and via collaborations. So now on a more personal note, so the grant, which is now we are in the final year, really led to, I think, to me, a conceptual shift. So beyond the papers and the future grant ideas and so on, it allowed us to think about microbiomes differently because we have these different perspectives. It allowed us to establish collaborations, and it's 3-4 years, but that's just the beginning. And personally, it allowed me to appreciate and understand the work that is done in other disciplines and in other domains. For example, just looking at Caitlin and Jamie doing these experiments, and it made me understand that it's really, really hard, and why it's difficult, and what are the challenges in other domains. And of course, explore new directions. So for example, in my lab, we're now predicting predictive ecology because it's essential to this kind of work. But we're not done, because as I said, it's just establishing. We have 3 major topics that are still in the works. And the last one of them is actually, if you want to learn more about it, you should go talk to Caitlin. It's really, really cool project about community response to environmental effects. So right after this talk, in poster number 18. Yeah, so with this, I will conclude.